Hello, everyone, and welcome to today's webinar with Dr. Suzan Imren, part of our Experts in Hematopoiesis series. My name is Amanda Fentiman. I am a scientific marketing specialist at STEMCELL Technologies, and I will be your moderator today. I am pleased to welcome Dr. Imren, who will be giving her presentation entitled Targeting Self-Renewal Function of Normal Hematopoietic and Leukemic Stem Cells. Dr. Imren is Senior Staff Scientist at the Fred Hutchinson Cancer Research Center, in addition to her role as a Clinical Assistant Professor at the Seattle Children's Hospital. She has been an author on over 10 publications related to her work on cord blood HSC expansion and work regarding in vivo models of human leukemogenesis. Just a note about the logistics of the webinar. If you have any questions, please type them into the Q&A panel on the right, and they will be addressed at the end of the presentation. Also, for your convenience, a link to the recorded version of this presentation will be emailed to you in about 2 weeks' time. Now, without further ado, I will hand over the microphone to Suzan.
Hello, everyone. In today's webinar, I'll give you some of the highlights of our efforts targeting self-renewal function of normal hematopoietic and leukemic stem cells. I will do this in 3 parts. In the first part, I will focus on how we can enhance the growth of normal HSPC, and I will summarize how we went beyond HOX to small molecules to UM171, and I will also tell you about progress that has been made with Notch ligand delta 1. In the second part, I will summarize some of the data that has been generated with gene-modified or edited HSPC. This will help to broaden the use of HSC gene therapy, and it will be important to prevent HSPC loss during ex vivo manipulation. And expanding gene-modified or gene-edited cells will reduce or even eliminate the use of myeloablative conditioning in HSC gene therapy setting. And as you can imagine, majority of these patients have non-malignant disorders. If myeloablative conditioning are eliminated, it will be a more attractive therapeutic alternative for these patients. And in the last part, focus on our efforts, how we can suppress the growth of leukemic stem cells and some lessons we have learned from MM1. Long-term repopulating cells, as you know, are capable of regenerating the lifelong production of all mature blood cells, but they show a delayed engraftment pattern. And these cells have extensive self-renewal potential, and they are multipotent, and yet have limited proliferation. Whereas short-term repopulating cells have limited or no self-renewal capacity, and they lineage restricted, and they can proliferate. Why do we bother to expand any of all those either long-term or short-term repopulating cells? And which are the target cells we should be focusing on? Which are the most key important cells for successful stem cell transplantation? Is it at all possible to do this? This slide shows that more patients are receiving hematopoietic stem cell transplantation for malignant and non-malignant diseases. And as of 2014, nearly 1,000 cord blood transplant has been performed, and this is overall 15% of all the transplantation done. The data has been acquired from NMDP website. And the availability of and stem cell transplantation becomes available based on the availability, safety, and efficacy of the donor source and stem cell numbers. Finding a donor is the major limitation of stem cell transplantation, and the ethnicity impacts this as depicted in this graph. The likelihood of finding an 8 out of 8 HLA match is shown here on this graph by the ethnicity. For white Europeans, it's very high. 78% of them will be able to find a match, 8 out of 8. And Native North Americans, it's 55%. However, for all of the other races, it's less than 50%. And as you can see in this table here on the right, the likelihood of finding a matched cord blood unit is more than 95% if the patient is less than 20 years old, and it's more than 80% if the patient is greater than 20 years of age. And for this reason, cord blood is being used more frequently as a source of stem cells. In addition, cord blood as a source of stem cells have several advantages. Better tolerance for HLA mismatch between donor and the recipient, and there is better graft versus leukemia effect, and therefore reduced risk of relapse post-transplant. And this has been recently published by our group, Dr. Filippo Milano, recently in New England Journal of Medicine paper. And cord blood is also a renewable source of donor HSCs. It's readily available, quick search time, expands the donor pool, has decreased incidence of chronic graft versus host disease. But on the other hand, there are also some disadvantages associated with cord blood as a source of stem cells. The low cell dose is the major one, and as a result of this, the hematopoietic recovery is quite delayed, and this exposes the patient to higher risk of early transplant-related mortality. And most of the time, in the first 100 days, these are due to infections. And the other one is this is a one-time donation. And if the patient needs donor lymphocyte infusion for relapse, this will not be possible. And also, the healthcare costs are higher with cord blood transplantation. The grafts are expensive, and intensive care admissions in the first 100 days post-transplants are more often with these patients. To overcome this issue of low cell dose, most patients now receive 2 units of cord blood to provide adequate stem cell numbers. Double cord transplant did overcome the risk of graft failure, but as you can see here on this slide, did not improve the neutrophil recovery as well as platelet recovery. They remained delayed. One can clearly see on this graph here on the left, on the right, that transplant-related mortality is closely related to the days achieved neutrophil recovery. And as you can see, patients who have achieved neutrophil recovery less than 26 days have significantly less, 19% transplant-related mortality, whereas it was 35% in patients who achieved neutrophil recovery greater than 26 days. We and others, while I was in Vancouver, in Keith Humphreys and Connie Eves' group, in collaboration with Guy Sauvageau, and now in Colleen Delaney's group in Seattle and others, have been very interested in the development of methods for expanding cord blood HSPC ex vivo for clinical applications. Therefore, we need to identify ways to intensify the self-renewal activity of normal HSPC. In order to do this, we need to better understand how HSC self-renewal is regulated. This slide is taken from a recent review by Goyama and Malloy, recently published in Blood, summarizes genes and pathways regulating human HSC function that have been identified in xenograft models. The interplay between the stimulatory signals as well as the inhibitory signals regulate self-renewal, proliferation, differentiation, and apoptosis of human HSPCs. I have underlined in red the modulators that I will be talking about today. My interest in human HSPC expansion started with HOX genes while I was in Keith Humphreys' lab in Terry Foulkes' laboratory. This slide summarizes many years of work, and many talented people were involved. HOXP4 we found out that HOXP4 is a powerful stimulator of mouse HSC expansion ex vivo. This was shown by Jennifer Antonchuk more than a decade ago when it was delivered as a trans-2 cDNA up to 40-fold, and also shown by Jana Krossel in Guy Savageau's group when it was delivered using a TAT fusion protein. And moreover, NUP98 and HOX fusion genes were even more powerful stimulators of mutant HSC expansion ex vivo, more greater than 1,000-fold. And this was also shown by Hideoki Ohta in Keith Humphrey's group in now a decade ago. However, the impact of HOXP4 on human cord blood HSPC expansion was less dramatic than what was documented for mutant HSC. And we saw a 4-fold increase in non-skid repopulating cells quantitated at week 16 after transplantation. In retrospect, that week 16 data was actually still a short-term repopulating cells. Using HOX-A10, just and only the homeodomain fragment, induced a up to 20-fold expansion of highly purified rhodomine low, lineage negative, 34 positive, 38 negative cord blood HSPCs. But again, the extent of this expansion was significantly lower than what has been documented in murine system. So, but as you can see here, these expanded cells underwent normal lymphomyeloid differentiation in vivo, and this was assayed in NSG mice 5 months after transplantation. Simultaneous studies done in collaboration with Hans-Peter Kiem at FATH at the time, using non-human primate bone marrow cells, suggested that differential effects of HOXP4 on short-term repopulating cells, and whereas NOP98 HOX-A10 homeodomain on long-term repopulating cells. So what we learned from these studies, that magnitude of expansion with these genes appeared suboptimal, and delivery of HOX genes as TAT fusion proteins remained a challenge due to their very short half-life in culture. And we needed alternate strategies to improve the system. So this slide summarizes the strategies that have been developed and tested clinically to overcome low cell dose issue in cord blood. They focus on using mediators that either enhance homing or induce expansion and proliferation or cord blood HSPC. And prostaglandins or CD26 peptidase inhibitors, sitagliptin, are some of the agents that enhance homing. Defined growth factor conditions, Notch ligand, delta 1, nicotinamide, SR1, copper chelator, UM171 are some of the modulators that have been used to expand and proliferate cord blood HSPCs. I have underlined both delta 1 and UM171 that I was involved in the studies that I will summarize in the next several slides. This figure was taken from a recent publication by Lon et al. In Nature Reviews. UM729 and later the synthesized molecule UM171 was discovered as a result of a screen of a library of 5,000 low molecular weight compounds for their ability to overcome the loss of CD34 positive and CD34 positive 45RA negative more primitive population mobilized peripheral blood in 7-day cytokine-supported culture. Screen was carried out in Guy Savageau's lab in Montreal. These are pyrimidinol derivatives. They do not downregulate aryl hydrocarbon receptor pathway. And at the moment, the mechanism is still unknown, but we know that they suppress genes associated with erythroid and megakaryocytic differentiation. While I was in Vancouver, we were very fortunate to be involved in the preclinical studies validating the impact of UM171 on ex vivo expansion of cord blood HSPC. This slide summarizes the data generated by Imam Fares in Guy Sauvageau's group in Montreal. As you can see, UM171 was tested for its ability to expand cord blood HSPCs using an automated and continuous medium delivery system over 12 days. And this system was developed in Peter Zanstra's lab at University of Toronto and was called fed batch system. This system optimizes the balance of stimulatory and inhibitory factors in a small volume. The results of the limit dilution experiments indicated that UM171 promoted a 13-fold expansion of human long-term HSCs in fed batch cultures of CD34 positive cord blood cells. And long-term HSCs were capable of reconstituting human hematopoiesis in the secondary recipients. And simultaneous addition of SR1 to UM171 did not change the frequency of long-term HSCs numbers generated at the end of 12 days, suggesting that these two molecules did not have any cooperative effect on long-term HSCs. On the left, you can see representative FACS profiles showing that expanded human cord blood cells are capable of reconstituting human hematopoiesis for at least 6 months in NSG mice, even when they have been transplanted with the progeny of as few as 100 day zero cells. Based on this very exciting preclinical data, Guy Sauvageau, Danny Claude Ray, and Sandra Cohen have initiated a phase 1/2 clinical trial. The study is currently recruiting patients in Canada. Nearly a decade ago, Colleen Delaney in Seattle had taken another approach to overcome the issue of low cell dose in the cord blood transplant setting. She explored the use of ex vivo expanded stem and progenitor cells to enhance the kinetics of hematopoietic recovery. This is depicted in this cartoon, in which the expanded cord blood cells are infused on day 0, unmanipulated cord blood unit. The expanded cell population is indicated in orange, is intended to provide the initial wave of rapid hematopoietic recovery, while the unmanipulated unit in blue follows its normal kinetics of recovery. This wave of early engraftment will result in early myeloid recovery and help patients to become transfusion independent quicker and be discharged from the hospital sooner. The blue line represents the normal kinetics that is seen in cord blood transplantation without the expanded product, with delayed engraftment, longer time to transfusion independence, and longer hospital stays. Colleen had focused on the use of engineered Notch ligand delta-1 to activate endogenous Notch signaling in cord blood-derived HSPC, which inhibited ex vivo differentiation and resulted in a significant increase in the absolute number of CD34 positive HSPC available for clinical applications. The role of Notch signaling pathway in hematopoiesis was discovered by Herb Bernstein's group in Seattle. Subsequent studies in his lab determined that the ligand of Notch1 delta-like 1 promoted ex vivo expansion of mouse and human hematopoietic stem and progenitor cells by activating endogenous Notch signaling. The delta-1 was engineered. Its extracellular domain was fused to the Fc portion of human IgG and had to be immobilized for its ideal activation and using fibronectin. And based on the preclinical data Colleen had generated with human CD34 positive, CD38 negative cells, she initiated a phase 1 trial using this platform. CD34 positive cells were enriched and cultured in serum-free media with 5 growth factors in flasks coated with fibronectin and an engineered Notch ligand delta-1. At the concentration, 2.5 micrograms per ml, that promotes proliferation of the progenitor cells. And the goal will be provide cells that rapidly engraft and overcome the delay in hematopoietic recovery. Ex vivo expansion schemas are shown in this graph here. In the initial phase 1 trial, the expanded cells from an HLA-matched cord blood unit were infused into patients at the end of harvest. CD34 cells were enriched. Negative fraction enriched in T cells were discarded. Cells expanded and harvested and transplanted to patients 4 hours after the infusion of another partially matched or matched cord blood unit. This method had many logistical risks for the transplant delay. Therefore, efforts were established to move to an off-the-shelf approach where you see here on the right, expanded cells were cryopreserved at the end of the harvest, and these cells were not HLA matched. And when needed, a unit was selected for each patient, thawed, and infused along with 1 or 2 unmanipulated cord blood units. Initial clinical trials showed that time to achieve an absolute neutrophil count greater than 500 cells per microliter was shortened significantly with a median of 11 days in the HLA match trial here in red, and to 19 days in the off-the-shelf trial. In the HLA match trial, the average CD34 cell dose was 12.5 10 to the 6 per kilo, whereas in the off-the-shelf, somewhat lower, but still 6.8 times 10 to the 6 per kilo. So this enhanced neutrophil recovery in both these patients were probably by giving a high dose of CD34 positive cells. Patients undergoing double cord blood transplantation with an identical conditioning regimen had a median time to neutrophil recovery of 25 days as seen here. Time to platelet recovery was also significantly shorter. It was 35 days in patients who received the off-the-shelf expanded product, whereas 43 days in patients who did not receive the product. Moreover, by day 50, post-transplantation, 92% of the patients in the expanded cohort had platelet engraftment, whereas in the conventional group, only 69% of the patients had platelet engraftment. Interestingly, we also noted that the risk of bacteremia after myeloablative cord blood transplant was significantly reduced with infusion of the shelf expanded cord blood progenitor cells. The median time to first bloodstream infection in the conventional group was 8 days, whereas it was 36 days in patients who also received the expanded product. And as you can see here on this graph on the right, significant less patients had documented infection compared to the patients who did not receive the expanded product, suggesting that rapid myeloid engraftment achieved with infusion of expanded progenitor cells protected patients from bacteremia. Colleen also wanted to see the clinical utility of the off-the-shelf expanded product in chemotherapy-induced neutropenia. And this was done in a phase 1 trial. And in patients who received intensive chemotherapy for AML, they were treated with G-CSF primed clofurabine and high-dose RSC. And 24 hours later, they received expanded cord blood progenitors during induction. And if they were qualified, they also received a second infusion of the expanded cells during their reinduction and consolidation. And you can see here on the bar graphs that chemo-related infection rates were significantly reduced with infusion of the off-the-shelf expanded cord blood progenitors in patients both during induction and also during reinduction and consolidation, both for documented infections that are the combination of suspected infections and also microscopically proven infections. And this was recently published in Lancet Hematology last year. We wanted to better understand the mechanism, how the expanded cells were helping the patients developing less bacterial infections. So Fabiola in the lab wanted to determine the functional properties of newly generated myeloid cells, first in NSG bone marrow and lungs, 7 days and 14 days after infusion of expanded or unmanipulated cord blood HSPCs. So in these experiments, mice either received expanded progeny or unmanipulated cord blood cells, and 7 and 14 days later, the bone marrow and lungs were harvested. Phagocytosis and oxidative burst were simultaneously detected in the bone marrow and the human cells in the bone marrow and the lungs. Fabiola did this by looking at the granulocytes and monocytes in the mouse bone marrow at day 7, and she incubated the cells with Staph aureus that was conjugated to a pH-sensitive dye within the acidic environment of the phagous lysosome. This dye would fluoresce and will be visible on PE channel, as you can see here. She did this with Staph aureus because the patients after cord blood transplantation, this is one of the pathogens that is more prevalent, but she also confirmed that these cells were also capable to phagocytose gram-negative bacteria E. coli as well as fungus. And in the same cell, she was also able to show oxygen-dependent killing as seen here because cells were incubated with a non-fluorescent dye, 1,2,3 dihydrorhodamine, that breaks down to fluorescent 1,2,3 rhodamine, and that is seen in the FL1 channel. And she could also detect some myeloid dendritic cells, and they also were capable of phagocytosing Staph aureus. But dendritic cells did not perform oxygen-dependent killing as expected. Fabiola was able to show that at day 7 after transplantation, in the bone marrow of these mice, there were significantly more human cells as seen here. And these human cells were capable of phagocytosis significantly more than the myeloid cells that were the progeny of unmanipulated cord blood unit. And moreover, the oxidative burst was significantly higher in these cells generated from the expanded unit. Interestingly, Fabiola was also able to show increased number of functional granulocytes and monocytes and myeloid dendritic cells in the expanded group mice in the lungs at day 7 post-transplant. And you see significant number of more human cells in mouse lungs and definitely more phagocytosis in these human cells in mice that have received the expanded cells. And this was done at day 14 due to the technical difficulties of accumulating human cells in mouse lungs at day 7 after transplantation. We then wanted to look at the functionality of newly generated myeloid cells in patient blood 7 and 14 days after infusion of off-the-shelf expanded cord blood progenitors. And these patients were enrolled in a phase 2 clinical trial that's currently ongoing. They were randomized upfront to receive expanded product along with conventional cord blood unit, and following a myeloablative conditioning regimen, which included cyclophosphamide, fludarabine, and TBI. 7 and 14 days after transplantation, flow cytometry was performed similarly to the mouse experiments, and phagocytosis and oxidative burst were performed. These are FACS profiles from a representative patient. As you can see, the patient who has received the off-the-shelf expanded product has significantly more granulocytes circulating in the blood 7 days after transplantation. And the patient that has received unmanipulated unit has remarkably less myeloid cells. And interestingly, these granulocytes in the patient that has received the expanded product significantly more competent and in phagocytosing staph aureus as seen here. However, interestingly, both of these myeloid cells that were able to phagocytose had similar potential for oxygen-dependent killing as seen here on the last histograms. And the slide summarizes the data from 6 patients that we have accumulated so far, that at day 7 post-transplant, significantly more functional myeloid cells were present in patient blood who were infused with off-the-shelf expanded cord blood progenitor cells. As anticipated, chimerism studies showed that at day 7, seen here on the left, at day 7 after transplantation are from the progeny of expanded unit seen here. And at day 14, the cells from the expanded unit are gone, and now they are the progeny of the conventional unit. But the frequency of phagocytic cells derived from expanded and conventional cord blood units are similar, as seen here on the right. In patients who have received the expanded progenitors, have many of these functional myeloid cells at day 7 after transplant. This is the first time we have looked, obviously, and probably these cells are beneficial to the patients and protecting them from infections early after transplantation. The conclusions in this first part, we and others have showed that cord blood engraftment kinetics can now be improved in patients through expansion of stem and progenitor cells ex vivo. Larger clinical trials are needed to confirm that enhanced hematopoietic recovery will result in decreased transplant-related mortality and morbidity from infectious causes and improve patient outcomes in terms of survival from malignancy. And the cost of cord blood units are substantial, and when double cord blood transplantation is performed, even higher. And the cost of manipulation of a unit could be also substantial. But if the expanded product could result in reduced hospitalization for transplant or a decrease in infection rates, antibiotic use, or less time spent in intensive care unit, may prove to be cost-effective. And also future studies or trials may consider combining complementary strategies that act through distinct mechanism and also on distinct population of HSPC. I will move on to the second goal, which will be to broaden the use of HSC gene therapy. As I mentioned before, by taking advantage of the tools developed in the first section, we will be maybe able to prevent the loss of HSPC during genetic manipulation, and then further expand these gene-edited or modified cells, and again, as I mentioned, and eventually reduce, eliminate use of myeloablative conditioning. While in Vancouver, we serendipitously found that UM171 enhanced gene transfer, as seen here, up to 2-fold after a quick exposure during pre-stimulation for 16 hours and infection for 6 hours. And also UM171, such short exposure, 22-hour exposure to UM171 enhanced the number of transduced cells up to 8-fold. Mice were transplanted with equal number of cells transduced in the presence or absence of UM171. At 30 weeks post-transplant, cells were transduced in the presence of UM171, outcompeted control transduced cells in vivo, indicating that short-term exposure to UM171 during pre-stimulation and transduction enhanced gene transfer to lymphomyeloid repopulating cells. And similar pattern was also observed in lymphoid and myeloid cells, suggesting that UM171 truly enhanced transduction and expansion of long-term lymphomyeloid repopulating cells. In the last section, I will review the approaches we have taken to suppress leukemic stem cell function. But to be able to do this, we need a model where we can better understand leukemic stem cell self-renewal. It was originally believed that transformed HSCs gave rise to leukemias. However, many studies have revealed that leukemia can also rise from transformed cells that are more mature, the progenitor compartment. With such heterogeneity in the source of disease, there's a great need to identify and better understand aspects of leukemia-initiating cells. And therefore, I became interested in developing a forward genetic model of aggressive human AML while I was in Vancouver in Keith Humphrey's group. Because this model will help identify cell of origin and many genetic and epigenetic hits required to generate LSCs, and will identify protein pathways, and also we will be able to find new therapeutic targets. And to do this, we took advantage of meningioma-1, a potent leukemogenic transcription cofactor, which overexpression was a poor prognostic marker in cytogenetically normal AML patients. And this gene was a cooperating partner with multiple genes, NUP98, HOXD13-1, AML1, or HOXA9. MM1 overexpression as a genetic hit was sufficient to induce AML in mice. This was shown by Michael Heuser while he was in Vancouver. And MM1 collaborated with NUP98, HOXD13, and D13, I will now on call, and massively enhanced self-renewal of mouse leukemia-initiating cells. And moreover, loss of MM1 expression impaired proliferation of clonogenic activity of cells from a human leukemia cell line. And so the objective was to investigate the transforming potential of MM1 in human cord blood cells. We were able to show that the forced expression of MM1 in CD34 positive human cord blood cells promotes proliferation in vitro on stroma-dependent cultures. During the 8-week culture period, the number of GFP positive cells increased by nearly 30-fold in MM1 transduced compared to controls. Similarly, at the end of 8 weeks, we found the culture initiated with MM1 transduced cells contained 300 more colony-forming cells. MM1 overexpression alters differentiation in vitro, as seen here. Significantly more common myeloid progenitors and granulocyte monocyte progenitors were generated at the end of 8 weeks in MM1 cultures. MM1 induced abnormal myeloproliferation in vivo. It was not sufficient to induce leukemia. Median survival of the mice was slightly shorter than the controls, but these mice never developed leukemia. Michael had previously shown that in mouse system, MM1 and ND13 cooperated to massively enhance self-renewal of the mouse leukemia-initiating cells. And therefore, in an effort to stimulate full leukemic transformation, I co-expressed MM1 and ND13 in cord blood cells, sorted these cells, and expanded them in vitro under stroma-free conditions. As you can see, both MM1 and ND13, also ND13 cord blood cells, showed enhanced growth in the first 2 weeks compared to MM1 and control transduced cells for up to 75 days. At the end of the cultures, there were significantly more primitive cells, 34 positive, 38 negative, or 34 positive, 38 positive in the MM1 and ND13 cells. Cells from these 1 to 3 month old cultures were then injected intrafemorally into NSG mice that is transgenic for human growth factors, IL-3, GCSF, GM-CSF, and stem cell factor. Strikingly, mice injected with MM1 and these 13 cells developed rapidly fatal AML with a median of 65 days, suggesting that MM1 cells are dependent on stroma support to sustain stem-like cells in vitro. Limit dilution transplants showed that the frequency of disease-initiating cells was thousandfold higher in MM1 and ND13 cells as compared to ND13 cells, demonstrating the leukemic synergy that can be obtained when MM1 and ND13 are co-expressed in cord blood cells. And as you can see, this AML was transplantable And this is a bone marrow slide from a secondary recipient massively infiltrated with blast cells. Moreover, gene set enrichment analysis revealed that human HSC and patient-derived LSC signatures were enriched in MM1 and D13 cells, consistent with their high leukemogenic potential. Furthermore, genes found to be hypomethylated in a subgroup of AML patients were also enriched in the gene signature of MM1 and D13 cells. In contrast, gene signatures specific for GMP and CBP alpha target genes, more favorable leukemias, were downregulated in MM1 and D13 cells when compared to ND13 cells alone. These data suggest that leukemic potential of MM1 and D13 cells, at least in part mediated through genetic pathways that govern AML pathogenesis and stem cell self-renewal. So we were able to show that MM1 and NUP98 HOX-D13 are sufficient for leukemic transformation of human cord blood CD34 cells. Leukemic transformation is associated with HSC and LSC gene expression signatures. And this model provides a new forward genetic model of aggressive serially transplantable human AML. In the last slide, I would like to acknowledge Colleen Delaney, Herb Bernstein, Shelley Heimfeld, Filippo Milano, Fabiola Maryam, Carrie Stoltzman, Emily Darbo, Emily Cox, and members of the cord blood program at Fred Hutchinson Cancer Research Center, and also Keith Humphreys, Connie Eves, and all the members in the lab at TerraFox Laboratory, and our collaborators from Montreal, Guy Savageau, Iman Fares at University of Montreal, Peter Zanstra at University of Toronto, Michael Huser at Hanover Medical School.
Thank you very much, Susan. So this concludes the presentation of the webinar. We will now begin our Q&A session by addressing some of the questions that have been submitted. So the first question, based on the frequency of long-term HSCs, the addition of SR1 to UM171 does not provide any increase in the frequency of long-term HSCs. Do you think this is because UM171 achieved the maximum self-renewal potential, or because both small molecules activate the same pathways?
Actually, both molecules activate different pathways, and there's some data suggesting that SR1 acts more on short-term replicating cells, whereas UM171 acts on longer-term replicating cells. So probably they're acting on different populations of cells. So that's why there's no sort of additive effect when both of them are added into culture.
How many off-the-shelf universal donor units can be prepared from a single cord blood unit?
This totally depends on the expansion potential. Some units expand more than the others, and the patients receive, there's a cutoff for total nucleated cells, so that's 10 to the 8th per kilo. And for CD34, it's 10 million per kilo. So if this unit has generated many more cells, and there are sufficient for 2 patients, that's divided. 1 unit goes into 1 patient.
Why do you think fresh, matched, expanded, and off-the-shelf products have different median engraftment times? Is it due to being matched or not?
Definitely. In the initial trial, patients, the expanded unit was matched, and also, as you remember from the table, the CD34 content was higher in this group than the off-the-shelf group. So both the LCL match and higher CD34 dose impacted the median neutrophil engraftment.
Can the MN1 and D13 animal model be used to test AML therapy, aka can it be used for in vitro assays or drug screening?
Definitely. It's a very reproducible assay. It's a very reproducible model, and the leukemia is very titratable. If you want aggressive leukemia, you inject more cells, and if you want a more slow progressing leukemia, you inject less cells. And also these cells grow in vitro in minimal cytokine support for a long time. So it will be also very useful for in vitro drug screening assays.
Can UM171 enhance gene transfer in CD34 positive cells from other sources like peripheral blood or bone marrow?
Yes, we looked at that. It's similar degree of enhanced transduction efficiency is seen in mobilized peripheral blood as well as human bone marrow.
Great. I think that finishes our questions. Thank you everyone for attending, and thank you again to Suzan for the great presentation. Thank you very much.